Hello, everyone, and welcome to today's broadcast, Viruses in Soils. What do we know and where do we go from here? It is presented by Laura Zinke, PhD, a postdoctoral researcher at the University of California, Davis. I am Judy O'Rourke of LabRoots, and I will be your moderator for today's event. We're delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I would like to remind everyone this event is interactive. We encourage you to participate by submitting as many questions as you want, anytime you want during the presentation. Just click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box that appears on the screen. Our speaker will respond to your questions via email. If you have trouble seeing or hearing the presentation, please click on the Ask a Question box to let us know that you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. Please join me in welcoming Dr. Zinke. I will now turn the presentation over to her.
All right, hi. Thank you for joining me today at the Microbiology and Immunology LabRoots virtual event. So today I'm going to be talking about viruses in soils. So to start, I wanted to show you a picture of one of my field sites. So the background of this title slide is Russell Ranch Sustainable Agricultural Facility side of Davis, California. And this experiment that's employed a bunch of different types of agricultural management and over the last 25 years. And we've been working out here to start to characterize some of the viral communities in soils. But before I get into the science, I wanted to do a quick introduction. So like Judy said, I'm a postdoctoral researcher at the University of California, Davis. I am focused on studying soil viral ecology in the newly established Emerson lab here in the Department of Plant Pathology. And since soil viral ecology is a relatively new field, I wanted to take the opportunity today to talk about why should we study and why should we care about soil viral ecology. So as part of this overview, I want to talk a little bit about the past of this field. So what has been done up until now to frame what we're doing currently. And then I want to talk about some of the present things in soil viral ecology, some of the really exciting developments that have allowed us to really start to develop this discipline as more than a niche discipline, but really a large part that we think will become as important as something like microbial ecology. And then talk about some of the linkages to between viral ecology and soil chemical cycling, including some recent work that's been done. And then I'll take the chance to talk a little bit about some of the work I've been doing here in my postdoc and some of the directions we plan on taking this work. So why soil viral ecology? That's something that I guess asked a lot. And there are a lot of reasons to study viruses in general. Starting with, the viruses are very diverse. So morphologically, they're quite diverse. If you look at this transmission electron microscope image or TEM image that's on the screen, this is showing just a range of viruses from a natural sample. And you see that you have everything from these long filamentous viruses to these much shorter or smaller viruses, but that have these really dramatic hexagonal capsid heads. So in addition to looking morphologically diverse, viruses are the most genetically diverse organisms on the planet. They go beyond, even some of the very diverse bacteria and archaea, where you can have maybe 50% genetic identity between strains. Viruses don't even have a shared genetic code. Some viruses are DNA-based, some are RNA-based, and then they have different single or double-stranded genomes within those types of genetic nucleic acid encoding systems. So another reason that we should study viral ecology is that viruses infect all domains of life. This includes humans, as we well know, especially with cold season coming up, but also viruses will infect bacteria and archaea, so all the prokaryotes as well. So what I'm showing you here on this slide is kind of a simplified phylogenetic tree of different bacteria and archaea from a sample site that we'll be talking about in a little bit. And then everywhere you see a red dot means that in this one study from one site, they recovered viruses that infect these types of bacteria and archaea. So there's a huge host range in terms of what viruses will infect. Another thing that's important to know about viruses is they're very abundant. So in marine systems, which have been very well studied compared to other environmental systems in terms of viral ecology, viruses are usually about 10 times more abundant than bacteria. So even though viruses tend to be really small, numerically, they're a huge reservoir of diversity on Earth. And then something that's near and dear to my heart, so something I'll be working on in my postdoc, is viruses, we think, impact soil chemical cycling. And there hasn't been a lot of work on this previously, but what I'm showing you here is kind of a simplified diagram of some of the major biochemical transformations in agricultural soils. So essentially, plants will take carbon dioxide, turn it into plant matter, and then this plant matter it gets put into the soil either through root action or through the plants dying and being incorporated into the soil. And then this organic matter gets broken down by microorganisms like bacteria and fungus, gets turned back into CO2, and then other microorganisms can take nitrogen from the atmosphere and put this into a form that plants can use. So we know that the microbiome in soils is critical to the functioning of the soil. But we also know that viruses can infect all levels of these communities. So what are these viruses doing to these communities? Are they, helping the communities? Are they controlling the community makeup? Are they hurting the communities? And is there a way that we can perhaps manage these to better manage our agricultural systems as well? And then I love this image. I think this is a really striking way to demonstrate one of the ways in which viruses can impact things like carbon cycles in the soil. So this is an image of a bacterium getting lysed open by viruses. You see the little lighter colored dots within the darker splotches. Those are the viruses that cause otherwise what were once living cells to lyse open, and then they release all their really nice, juicy nutrients into the soil. So this includes things like sugars and proteins and nucleic acids and things that other microbes and plants might want to take up and consume. Something else that's really intriguing about viruses is that they don't always immediately kill their hosts. So this little cartoon is from a review from 2012 that's demonstrating that viruses can take over their host metabolism and tell a host, like a bacterial host, I want you to do this process. And they can alter what things like bacteria or fungus are actually doing in the soil. And then economically, something that's really important about understanding viruses in soils is their impact on crop management and crop disease. So I'm showing you here a picture of soil-borne wheat mosaic virus. So this is a soil-borne virus that has a slime mold vector. And this virus can live inside its slime mold vector in soils for up to 30 years, even after you removed the wheat from your soil. So understanding the baseline viral community and then also how this community propagates and survives in soils over long time periods is really important when we're trying to do things like develop better cropping systems or better land or soil management in our agricultural sectors. So something I want to emphasize is knowing that viruses are in soil is not new. We've collectively known that viruses live in soils for decades. I've just captured snapshots of a few different papers from multiple decades in which viruses have been previously studied from soil. And you see these go back to even literature reviews that came out in 1950. But if you also notice just from the titles, a lot of studies that looked at viruses in soils focused on either something like just kind of general viral transport in soils, or something like a specific virus, like, a specific plant virus or a specific virus that could infect humans. And, there's a reason for this. It's not that people haven't been interested in general viral ecology in soils. It's that it's been really difficult to study these systems until more recently. So some of the complications in studying viruses in soils are actually really closely related to some of the complications in studying microbiology in general. So here I have a little diagram of bacteria and viruses in soils. So the bacteria are the oval shapes, viruses are the hexagonal shapes. And there are a lot of viruses in soils. Some viral counts put these at maybe 10 to the 9th viruses per gram of soil. And a lot of us are familiar with the idea of the Great Plate Count anomaly. So that's the idea that if you, for instance, take a gram of soil and you try to put it on normal plate represented by the tannish circle here, then you'll get maybe 1% of your microbial component of the soil actually able to grow on the media that we give it. So that's represented by these blue ovals that have been able to grow on this plate. And to study viruses, we need to be able to have their hosts so that we have something for the viruses to infect and grow in, at least using a lot of traditional viral methods. So for instance, if we're only growing 1% of the bacterial population or only these blue microbes, then we can only study the viruses represented by the pink hexagons. So what that means practically is we're missing all of these viruses in the soil. So we have a very limited view of what viruses are actually present in soil. Another reason why so far we haven't made a lot of progress in trying to figure out what viruses are present in soil is that there's no universal marker gene for viruses. So a lot of huge leaps have been made in more microbial-based studies, like human microbiome studies, using targeted sequencing. So sequencing something like the 16S rRNA gene for bacteria or archaea, or the 18S rRNA gene for eukaryotes. But with the bacteria, with viruses, since they're so diverse and they don't even have always the same nucleic acid that they use as their primary mode of genetic recording, we can't use these sort of methods. So it's until recently, sequencing on a community level has not been very easy for viral ecology. So what have we been doing in the viral ecology field until now? So one of the things that's been really a workhorse for viral ecology is using microscopy. So the image on the left, the one with the green splotches on the dark background, that is an epifluorescent microscope image of a mixed viral and bacterial community. So you see that these bigger, brighter green things are prokaryotes, and then in the background, these smaller green dots are viruses. So this is one way that we've been able to tap into and study viruses by counting and comparing the number of viruses between samples. And then on the right, I'm showing you again this TEM image in which you can identify the morphology of different viruses. So you can get a qualitative estimate of the different types of viruses you have at a very coarse resolution. And then from these sorts of methods, the type of data you get back looks something like this. So it looks like, for instance, on or for the eplofluorescence microscopy, you can compare how many viruses are in each sample. So this bar chart on the left is showing that in some of these systems, so you see to the right are these EVES and MTPK sites followed by ag. So those are agricultural sites that tend to have fewer viruses than some of these undisturbed forest sites to the left of them. And then the images on your right are data that results from collecting these different morphological data using transmission electron microscopy. So you can compare, each bar represents a different morphotype or type of virus. And you see that generally in most of these samples, you have one type of virus, in this case, the Podoviridae are the most abundant, but then in the bottom right-hand corner, there's some difference in that, and we see that a different type of virus is more abundant. So this is pretty much what the field depended on for about 20 years to study viruses in soils. And a recent really fantastic review that came out in 2017 collated results from all these different viral ecology studies and looked at the numbers of viruses that were found in each different type of sample. So along the x-axis, there are the different types of soil ecotypes that were found amongst a review of these studies, including things like agriculture and wetland. And then on the y-axis, there is the order of magnitude range of number of viruses in these soils in grams per dry weight of soil. And we see that the natural systems seen in forests and wetlands tend to have the most viruses, but also things like cold deserts and agricultural systems have a fairly high number of viruses compared to something like hot deserts, which might be expected since that correlates pretty well to the number of bacteria we expect to see in these sites. And then the study also looked at the number of bacteria versus the number of viruses that were counted in each site. And remember, in these better characterized marine systems that we use as reference a lot, viruses tend to outnumber bacteria about 10 to 1. But in soils, that apparently doesn't necessarily hold. So for instance, in some systems, viruses dominate over bacteria, outnumbering bacteria up to 1,000 to 1. Versus in other systems, bacteria dominate, outnumbering viruses by orders of magnitude. And there's not an apparent trend between some of these systems. So for instance, in these orange bars, which are these deserty systems, there's quite a bit of spread versus agricultural systems where in some cases it appears that viruses dominate and some bacteria dominate. So what this is really telling us is there's a lot we don't know about viral dynamics in soils, and there's a lot of work left to do to try and understand what role viruses are playing in these different soil systems. And something that's been really helpful in viral ecology recently is DNA sequencing. So we know DNA sequencing from a slew of studies. It's really revolutionized the whole field of biology. And viral ecology is no exception. So something, I mentioned earlier that DNA sequencing for a specific gene is not going to work very well if you're trying to capture a whole viral community. But with the advent of untargeted sequencing, or in other words, shotgun sequencing of all the DNA in a sample, this has really been useful in viral ecology, but this hasn't been applied much as of 2017. So this table I'm showing you is, again, from this soil virus review from 2017, where as of that point, the authors had only found 6 different studies that had employed this shotgun or this broad sequencing to specifically viral communities. So the type of data we can get out of this shotgun sequencing is in some ways similar to the type of data we can get from using microscopy. So you can see different numbers of different morphotypes based on their DNA sequences. But what's really exciting and what I've been doing is you can take this further, and using all of this data you get, you can start to apply statistical techniques that are commonly used in ecology, both at the macro scale and used in more microbial studies. And then you can really start to tackle questions at a much finer scale resolution than comparing explicitly the number of viruses between samples. So one of the first times this was employed was in 2007. And this is work that was done by Noah Führer. And he found that from sequencing 3 soil samples and comparing them to some of the few other specifically viral sequencing efforts at that point, soil samples were unique and distinct from other environments. So this was, I think, surprising. I would have expected soil to be more similar to seawater or marine sediment than seawater or sediment are to the human gut. But in light of more recent findings in the microbial ecology field, we're starting to see that soil is actually this really different environment that has a whole slew of different factors that go into viral ecology that make it distinct from environments that are better characterized. So that brings us to today. So currently we're in what I'm calling the renaissance of soil viral ecology. So what does that mean? In addition to this becoming a really, broad field where you have a lot of different groups understanding, finally, viruses are, we can do things that make this a really robust science. So we can do a lot more samples at once. So instead of comparing maybe 3 different soil samples like we saw in the 2007 study a couple of slides ago, we can now sequence hundreds of samples at once. And we can also do a much higher depth of sequencing for each sample. And so what this means is that instead of sampling a specific virus or viral genome maybe once or twice in a sample, we can sample it essentially hundreds of times and become really confident about what we're looking at. So what these 2 points mean is that we now have more spatial and more temporal resolution. So instead of being able to compare soils from different states, maybe we're now starting to compare soils from within a couple of feet of each other, or like what I'm doing where we compare soils from different parts of the same cropping field. And we can also start to study temporal dynamics. So maybe that might explain some of these large variations we see in the bacteria ratio versus the viral ratio. And then this higher depth of sequencing per sample means that we can begin to recover complete viral genomes. So instead of getting a little snippet of DNA and trying to draw conclusions based on that, which is sort of analogous to getting one piece of a puzzle and trying to figure out what the whole puzzle looks like, we can now recover these complete viral genomes and be able to say, I think this is what this virus is doing. I think this is who this virus might infect. And I think that we can draw these conclusions based on these other factors and compare it to the environment. So to highlight some of these recent developments, I wanted to go through a study that was published last month in Nature Microbiology. This is a study done by Joanne Emerson, who's my postdoc advisor, and some of her collaborators from the ISOGenie project, which has been looking at thawing permafrost. So as a quick background, permafrost is permanently frozen ground, but with climate change right now, we are having significant thawing of permafrost. And this hasn't happened in a long time, so we're not sure what to expect from this. Are we going to see increased methane emissions from these sites? Are we going to change the carbon cycle on Earth? Things like that remain open questions. So one thing we wanted to do, or the authors wanted to do, was to study the viral ecology of this and see what role viruses are playing. So the map here with the red arrow is showing where the study was done. So you see this is in northern Sweden, called Stordal and Meyer, and the image on the bottom left-hand side is the kind of a panoramic view of this study site. So you see that this doesn't look that cold right now. It has some beautiful trees, a lot of lakes, and some areas that are kind of boggy and thawed. So a close-up view of this shows where the samples were taken. So what the authors did was they went to this thawing permafrost area and they took soil samples from different areas that have been thawed to different amounts. So for instance, all these brown dots you see towards the top of the picture are from what's called palza. That's the most frozen type of soil. And then there's a thaw gradient up till you hit fen, which is more just kind of like watery but fairly thawed soil. And by taking soil samples, they were able to collect 201 metagenomes from this thawing permafrost soil. So this means that they collected samples, they extracted all the DNA from the samples, and they sequenced them using a variety of, in theory, unbiased methods. And then once all of these metagenomes were sequenced and there was DNA ranging from mosses to bacteria, then these metagenomes were bioinformatically mined for viruses. And so, in other words, viral genomes were retrieved from these total soil genomes. And from this, the authors recovered almost 2,000 complete or near complete viral genomes, which is a huge number. But only 17% of these viral genomes could be taxonomically identified. So what that means is only 17% of these viruses are related to something where we know what the virus does and we have studied this virus somehow, usually through cultivation-based methods. So if you look at the pie charts on the bottom of the slide, you see that the green wedges are the taxonomically classified viruses. This total 17%. Most of the viruses are either unclassified but have been seen in other environments, typically in other soil environments, or have never been seen before, and this is the first time that this sort of virus has been recovered from a sample. So we see that there's a lot of soil virus diversity, and we don't know what these viruses are doing or why they're there, essentially. So then, using some different bioinformatic techniques, including looking at CRISPR intergenic sequencing spaces and where viruses were if they were inside of a host genome, as in they'd integrated themselves into the genomes. The authors of the study were able to connect the viral portion of the community to some of their hosts. So this is, again, this phylogenetic tree of all the bacteria and archaea that were found in these samples. And where all these red dots are where viruses that probably infect these microbes were found. And the numbers next to the dots show how many viruses were found that infect these hosts. So for instance, to the far left, you see the proteobacteria with the number 148. So this is a really common type of bacteria, and a lot of viruses apparently infect these bacteria. And so we can start to determine not just how many viruses are there, but also how they're impacting the microbial community. And in total, 35% of the almost 2,000 viruses that were recovered were able to be linked to a host, or in other words, to something that, like a bacteria, they'll infect. And the hosts included things like methane cycling organisms, including ones that will produce or consume methane, and also heterotrophic organisms who do the bulk of organic matter cycling, organic matter breakdown in soils. So this is exciting. We're seeing that potentially by infecting hosts who do really critical chemical transformations in the environment, the viruses could be impacting how hosts are cycling these chemicals. And then there was also a finding that viral communities from these different types of permafrost thawed soils. So the more thawed, like the fen in blue, versus the least thawed, like the polza in brown, are actually distinctive between the soil types. So these were compared to some of the different other parameters, like who the hosts were, or what the pH of the soils were, or how much moisture is in the soil. And there is a significant correlation found between some of things like the host community, or the bacteria that the viruses are infecting, and the viral community. So that's linking these viruses and who they infect. And also excitingly, it was found that viruses are corresponding to pH, which is something that a huge meta-analysis of all the bacterial communities in soils that we've seen found with bacteria. And we see that also pH not only affects bacteria, but also viruses. And then the last point I want to highlight from this paper is that viruses encoded glycoside hydrolases. So glycoside hydrolases are these enzymes that will break down organic matter, including organic matter produced by plants. So some of these glycoside hydrolases include enzymes that will break down different parts of plant material. So everywhere you see a red arrow next to these hexagonal cartoon sugar structures is a place where a virus-encoded hydrolase will cleave. So what this means is that viruses are also helping to break down organic matter in these thawing permafrost soils into smaller sugars that then the bacterial archaeology can either until it eventually becomes something like carbon dioxide or methane. So really what this study showed that viruses are impacting nutrient cycling in permafrost soils. But building on from that, I want to know how viruses are impacting nutrient cycling in agricultural soils. So located in kind of inland Northern California, I'm in an area where there's a lot of tomato farming, and that's something that UC Davis is really good at, is growing tomatoes, especially in long-term experiments and things like that. So I have this really great opportunity to sample soils and study virus communities from soils that have been maintained organically for crop production for 25 years, as well as soils that are maintained in kind of your conventional or industrial agricultural settings, and biochar amended tomato field soils. So what biochar is similar to charcoal. You can take things like waste products from agriculture, like almond shells or walnut shells or pine needles, and you can heat these up to evolve the chemistry of these materials. And then what's been evolved, some of the gases can be used for biofuels or biogas, and what you're left with is essentially charred or ashed organic matter. And this can be buried in the soil, and this will do a couple of things. This can be used for carbon sequestration, so putting carbon back into the soil in a form that will probably not be broken down very quickly. And it can also be used to retain soil moisture or nitrogen. But there's still a lot of uncertainties in terms of what biochar or how biochar interacts with soils, including within the living components of soils. So that's something that we're actively studying right now, is looking at soils from different types of biochar amendments. So biochar that was made from different feed sources like almond shells versus pine needles, and how this is impacting the viral community. So to do this, what we do is we start out going out to the field. So we sample soil from tomato fields, and I'm showing you a couple of pictures of our sampling back in July, where we had one of our fantastic undergrads come and help and let us sit back and watch her do the dirty work. And so that's the picture on the left, and then the picture on the right, I'm holding up a bag of freshly sampled soil. So then once we get the soil back to the lab, we do a couple of things to it. We still quantify, so we still do counts of viruses under the epleifluorescent microscope to try and figure out how many viruses are there. So how we do that is we separate the bacteria and the viruses from soil. And then we take this separated bacteria and virus-full liquid and we pass it through a 0.2 micron filter. So what that does is that filter will allow viruses and other small particles to be separated from the larger cellular community, like the bacteria community. And then we can centrifuge this, we get exclusively viral particles, and then we can stain these with a nucleic acid stain and put them under the microscope. And we get something that looks about like this. So this is an image from our two fantastic undergrads who have been working on trying to make these microscopic counts really robust and repeatable. And interestingly, preliminary numbers are showing we're getting somewhere around 10 to the 7 viruses per gram of soil, which is a little bit on the lower end. But this is also sampled at a point kind of later in the season. So this soil is a little drier. There might not be as much living in there. So this is exciting. This is starting to really allow us to connect the viral counts, or how many viruses are there, to the actual viral ecology. And then what I'm focusing on in my postdoc is taking these viruses and further concentrating them so that we can extract their DNA. So what this looks like is ultracentrifuging viruses to get them out of solution for several hours and extracting their DNA and sequencing that, as well as we look at and sequence the total DNA of the soil. So this includes the bacteria and the fungus. And then we run all of the sequences through a bioinformatic pipeline that looks about like this. So once you have DNA and you sequence it, you get back, using the technology we use, which is Illumina HiSeq sequencing, we get back DNA fragments that are about 125 to 150 base pairs long. And that's pretty short. That's not always super informative at that length. So what we do to make more sense of this data is we will assemble these fragments. In other words, we'll try to reconstruct genes from these smaller pieces of DNA. And then from these reconstructed genes, we filter out the sequences that are too short to be confidently called as viruses. And then we do another check to ensure that all of our sequences are from viruses. And we use a program called VirSorter to do that. And once we have all of our sequences that we think are viral, we can start getting into the real science of it. So you can ask questions like, who are these viruses, or who are these viruses infecting? We can ask questions about their function. So what are these viruses doing? Do they have these extra metabolic genes to tell their host to do something? For instance, with the glycoside hydrolases that were found in the permafrost study. And we can start to look at relative abundances of viruses. So are a specific type of virus more abundant during the summer when fields have tomatoes actively growing, or are they more abundant during winter when there's maybe not anything growing in that field? And some really preliminary work that we have from some of these tomato field soils that have biochar in them is that there are quite a few viruses in these soils. So I'm showing you here just a quick bar chart with the different types of soil amendments on the bottom. So this includes a control that's just normal tomato soil, or the normal soil that we plant tomatoes in here in Northern California. A type of soil that's been amended with charred coconut shell, pine, or almond shell. And what we found from this is that the number of viruses is high, and it's actually quite diverse. So we recovered over 3,000 viral populations from over 6,000 viral genomes. So what that's saying is we recovered about 6,000 viral genomes, and then about half of those are unique within the data set. So in addition to taking that study further and really delving into who those viruses are and what they're doing, some of the other work I'll be doing in my postdoc coming up in the next year is comparing viruses between organically managed and conventionally or traditionally managed soil. So in conventionally managed soil fields, you usually get some sort of fertilization from like a liquid mineral fertilizer. So you get your nitrogen in the forms of like nitrate, ammonium, like inorganic forms. And pesticides could be used for pest management. Versus in the organic soils, the fertilizer all comes from chicken manure or from a cover crop that's planted in the winter, like a legume that will put nitrogen back into the soil. And some of our collaborators have already shown that this will change the microbial community. So we're interested in, does this change the viral community, and does this have any impacts on the functioning of the soil? So how, in general, chemicals are cycled in the soil, or in terms of how this could impact plant health. So are there viruses that can either promote plant health by, say, killing plant pathogens, or are there viruses that are pathogenic to plants residing in the soil? And then within this, we'll also be comparing seasonality of soil viral communities. So do these communities change with this season? And we suspect that they do, but stay tuned. We'll get to that. And that is all I have to say for today. And I really want to thank you for your time listening. And I also want to thank some of my fantastic lab mates and collaborators, my PI, Dr. Emerson, our at UC Davis, who have been responsible for getting all the biochar fields set up and running.
Thank you, Dr. Zinke, for that informative presentation. We will now start the Q&A portion of the webinar, and we'll address some of the most commonly asked questions by our viewers. If you have any questions you'd like to ask, please do so now. Just click on the Ask a Question drop-down box located on the far left of your presentation window. Type your question into the box that appears on the screen and click the Send button. Our speaker will follow up with your questions via email. So let's get started. Our first question is, what are some of the major limitations in viral ecology, and how do you address those?
Yeah, so like any field, new or well-established, there are several drawbacks that we really need to consider when doing our studies. I think one of the most major ones is a problem that we also have in microbial ecology, where through studying anything without culturing it, we're dependent on things that have been cultivated to understand what they're doing. So as long as we have, for instance, a gene that's been previously studied, then we can compare what we find in our novel systems and compare to what's been previously studied and say, okay, we think that this is doing this because they look similar or they have similar sequences. But we also get a lot of DNA sequences where we have no match to them. And those are going to be really hard to figure out what they're doing just from looking at a DNA sequence. So that's when we have to go back in to our samples and try and recover those and actually culture those. So that's, I think, one of the biggest drawbacks right now is we're really limited in terms of what we've previously studied for comparing that to what we currently are studying. And one of the other big drawbacks that I'm having with my work is that since viruses are so diverse in terms of if they're DNA-based or RNA-based and they have different double or single-strandedness, it's hard to, in one study, target all those different types of viruses. So I'm focusing mostly on DNA viruses right now, but for instance, a lot of plant viruses are RNA viruses. So that's something we're actively working on being able to simultaneously examine, but there's still some methods development that's going into that.
Well, thank you for that. Next one is, what are some of the applications of soil viral ecology?
So I think there are quite a few. I think they're all a little bit, maybe a decade off in the future. But some of the things that people are doing now that's really exciting is trying to use viruses that occur naturally to control plant pathogenic viruses pathogenic bacteria and fungi and things like nematodes and smaller or naturally. So virus that does not impact a plant or a human, and introducing it into an environment to infect a fungus that does negatively impact a plant is something that's preliminarily been really successful in some sectors. And there's a lot of evidence that we can also do that with things like mosquitoes or with other pests, including human pests. And that's why I think it's really important to keep studying viral ecology, since to do something like that, we also need to know what are the other viruses in the system and how we'll be impacting a whole system, so we don't accidentally, start a domino effect or anything.
Very interesting. Thank you for that. Next one is, are viral communities in soils positive or negative for the environment?
Yeah, so that's something I actually get asked a lot. So we have this view including from a bunch of really entertaining movies, that viruses are bad. When you think of virus, you think of something like the cold or the flu. And in humans and pathogens, those viruses are bad. But I think what's going to happen with our impression of viral ecology is it's going to be similar to what's happened in the past 10 years with looking at bacteria, for instance, in the human gut or in agricultural soils, where we know that there's a small portion of these entities, bacterial or viral, that aren't good for us or for plants or something like that. But actually, there are probably a lot that are really beneficial and critical to something like nutrient cycling or controlling populations of bacteria or other organisms that keep something from, blooming and going out of control. And yeah, so I think, it's definitely going to be dependent on the situation and the virus and what your application is. But I think in general, viral populations are a really beneficial thing to have in the environment.
Very good. We have time for one more question. Do human activities impact soil viral ecology?
Yeah, so we don't know yet. From some of the preliminary studies, so for instance, the graphs I was showing where there's different numbers of viruses in different types of soil, that seems to indicate that in some cases we might decrease the number of viruses present in some of the soils. So it looks like we could have an impact on that. We also know from other systems, so from things like sewage or wastewater treatment, that humans can help to increase the number of viruses in a given place. But again, that's something that we're not totally, and hopefully something that through some of my work, as well as the work of a lot of other really fantastic scientists.
Well, thank you. I would like to once again thank Dr. Zinke for her presentation. I would also like to thank LabRoots for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through December 13th, 2018. You will receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's event. That is all for now. Thank you for joining us. We hope to see you again soon. Bye.